Good afternoon to everybody. It is a really great pleasure to have one of the most impressive scientists I have met in my life, if I'm not exaggerating. I'm sure you will agree with me very soon. And a very good friend, Nikos Hatzakis, who has a chemist background, but he's doing absolutely fantastic innovative things from biology to drug delivery systems in a single molecule tracking strategy. A very innovative work. I don't want to waste more your time because Nikos is here. And Nico, the floor is yours. Thanks for joining here at seminars. It's a great pleasure to listen to your new work. Thank you.
Thank you, Yanis. Thank you, everybody. And imagine if we can observe biological events in real time. And today, that would be a way to evaluate all the elements that allow us to control biology. And today I'm going to show you this, the direct observation of biological events with an emphasis on neuronal cells and CNS in general, and how we can use AI to leverage this information and to utilize this to control biological events. So I'm the Department of Chemistry, University of Copenhagen. I did my sabbatical in HMS, and I'm a visiting professor for these 2 years. And I'm directing a center which is for optimized oligo escape. This is funded by the foundation of Novo Nordisk, and also a center for 4D cellular dynamics. And because I have a lot of free time, I also founded a company that utilizes this technology to help and bridge frontier science with industry and firm. And I'm going to start by, because you probably saw the title of 4D biology and you're like, what is this? We call 4D 3D over time. The fourth dimension is that of time. So in this movie that I'm going to show you is the direct observation of synuclein fibrils preformed as they enter in neurons. And each of the dots that you see over here is a preformed fibril of synuclein as it enters the cell. And in the beginning of the talk, I'm going to show you how we can observe this, quantify this, understand what controls their escape and the growth of synuclein fibrils. And then I'm going to show you some information, some of the latest results, how we can expand this to deliver antibodies, therapeutic antibodies, or lipid nanoparticles with therapeutics. All right, so when the movie plays, you will see what we really mean with 4D biology. Now, I'm going to bring myself really inside the cell. I'm looking up and I can find the particles. Each of the dots that you observe over here is one of the preformed fibrils that we deliver inside the cells. And then the second line that you observe as the movie plays is that you observe some lines. And these lines is what Google Maps does to you when you're using your phone or your Apple or whatsoever utility you're using your phone, because it can track your X, Y, coordinates and know at any point where you are. So these lines that you see over here is the pathway of the particles of the preformed fibrils as they enter the cell. And when you look at the data, and you will appreciate 2 elements, 2 layers of heterogeneity. The first layer of heterogeneity is the brightness. This guy is very bright, this guy is a little bit less bright, and these guys over here are less and less bright. And this brightness, we take this, we convert this to number of photons, and we convert this number of photons to copy numbers of individual proteins that you have in this fibril, in this nanoparticle, in this antibody. So you have a complete quantitative assessment of how many proteins you're looking. The second element that you observe is these lines, and these lines are highly heterogeneous. Some of them are pretty stuck in the same spot because the particle is trying to enter the cell, but is still stuck in the endocytosis. It hasn't really managed to enter the cell. And there are some other particles. Let the movie reach the point. And you can see these particles here with longer lines. They are inside the cell and they are moving through the endocytotic pathways. So of course they're moving longer and they have longer, as we call it, receptors. And at the bottom, you see some of the publications from my group that we can extract this layer of information. So now that you saw what we can do with the microscopy, we can go on the real data, on static data, that we can show what's downstream of this effect. So now we took this cell, this is a neuronal cell, we have fed it with preformed aggregates of synuclein. We have inside the cell EGFP, normal EGFP expressing synuclein. So when the fibril is being growing inside the cell, I will see it as dots. This is a static movie, but the beauty of this microscope is that it allows me to go through the action of the neuron and observe all this process where the synuclein fibers are localized, how many of them are they, how big are they, and all these layers of information that you want to extract. So as I write on top, is one cell, one aggregate at a time that this technology allows us to extract. So I'm going to start with a publication that came out a year ago. And this is in a very tight collaboration with the group of Tom Kirchhausen, who is a collaborator, is part of the center that I direct, and is also a very good friend. So, and this paper came out in 25, and it's driven by the group of Tom on the imaging, and we did the analysis. And I will show you how we took up this imaging in my lab later on. So we would take preformed fibrils of synuclein, and we could add them on cells, and the cells, neuronal cells that you can see here, were expressing the EGFP synuclein. So when you have not add preformed fibrils, you can see the EGFP of synuclein is perfectly soluble inside the cell. It's heterogeneous between cell to cell, but you can see everything is in the cytosol equally distributed. When you start adding the preformed fibrils of synuclein, now you can see that you lose this kind of homogeneous, though some of it still exists, but now you see some clustering at certain parts of the cell, and this is where you actually start having the whole formation of the first form of aggregates of synuclein. So we're very interested in this, and we were trying to see where does it localize. Is it like found primarily on the early endosome? Is it on the late endosome? Where exactly do we see these fibrils? And most importantly, what is the mechanism? Because we deliver the fibrils inside the cell, and then somehow inside the cell, these fibrils are starting growing, these preformed fibrils are starting forming the nucleation point that the fibril starts growing. So where is this happening? So what we did is that we did stable cell, this slide is years of work, obviously, because you have to have a stable cell line expressing early endosome marker or late endosome marker as the anti-lab over here. And these are stable cell lines, and we know that these things don't affect each other instead of using just transfection. On the left, I'm adding PFF, and I have the early endosome marker, and you can see these nucleation points are not colocalized. So you can see every blue point, that is the preformed fibrils, do not seem to colocalize, not to overlay with the data that you see with what correspond the early endosome. But when we start looking at the late endosome, you can see that this dot example, for example, that is a preformed fibril, is exactly at the same spot that we have late endosome, and this as well, and this as well. We also did exactly the same experiment with labeling one color the preformed fibril, a second color the synuclein, and the third color the early or the late endosome marker. And you can actually see here, I'm not going to bug you with all the details, you can see that the colocalization is massively increased. Is massively increased. Probably, and as you can see here, this very clearly states that the LAMP1 colocalization is 3 to 4 times more than the A1, or if I'm going to make it even more simple, that as it enters throughout the cell, we have the maturation of the endosome from early to the late, and I see everything or a higher extent colocalizing to what we call late endosome. And that actually shows a little bit some idea of where the synuclein colocalizes and starts aggregation, but it doesn't give us a very big information as to how and why. What is the mechanism out of this? So then we thought, because we work on this Center for Optimized Oligo Escape, and the whole idea is that you have that the endosomes open up and deliver genetic material. That's how viruses enter their cells. That's how lipid nanoparticles with mRNA, or if I call it vaccine, deliver the genetic material. And we question is like, could it be that the endosomes rupture, open transiently, and when this opening happens, then you can start forming the seeding of the aggregation. So then we took a marker, which we call galectin marker, and this galectin is a protein that senses oligosaccharides that exist in the inner of the endosome. So when the endosome is sealed and spherical, then the galectin will bounce. It will never find the oligosaccharides inside, so it will never colocalize. It will never bind for a long time inside the endosomes. But if the endosomes rupture and open up, the galectin will enter and it will localize there. And so you can see here galectin signal in the cells without preformed fibrils, and they are everywhere. When I start adding preformed fibrils, as you can see here, you see that the galectin signal is no longer diffuse all over the cell, as you can see here or there, but it starts now localizing in certain compartments. And if you can see a little bit carefully, you can see that the galectin here localizes very well with the compartment that is the synuclein, actually providing a mechanism that's showcasing that wherever you have galectin, which is a ruptured endosome, and this ruptured endosome is actually localized with where your preformed fibrils of synuclein is. And you can see the colocalization is profound here. So we have a high number of cells, and we can see that the effect is very clear. And then we were like wondering, okay, so now all this data, guys, and I need to stress this, are coming from these movies I showed you. So you can imagine the technology that you need to identify all this and colocalize all this data and extract this information from particles that are moving in the millisecond time scale. So then we question, all right, but if galectin is there, how is this happening? Because we know that endosomes, as they mature, they start from pH 7.4, and as they move inside the cell and they mature, the pH gradually, slowly, and gradually stops until it reaches what we could call in the range of 5.5 or something for the late endosome and the lysosome. So we were like, if the if the endosomes rupture, then probably we should be able to measure that at that endosome that opened transiently, the pH should have neutralized, because you open up a hole and the pH becomes exactly the same as in the cytosol of the cell. So we made an assay that actually now uses pH sensitive reporters. And in essence, I'm not going to kill you with all the details, though I'm extremely happy to discuss all this because that's a big part of the analysis that we do. We put two chromophores in there. One of them is pH sensitive, the other one is pH insensitive, and we measure the ratio of these two chromophores at different as the particles move in the three-dimensional space, and we have constructed the calibration curve of the ratio to convert this to pH values. So a certain ratio corresponds to one pH value. When the ratio changes, it corresponds to another pH value. So this allows us, however simple or tragic or challenging it may sound, that we can do this for hundreds of particles as they move inside the cell. And this is the distribution of ratios and pH that you can see in these cases. And you can actually see, and I think the most important thing is that in the absence of presence or of papilli mode, you can see that there is a small variation in the pH. And most importantly, when we added small molecules, this is papilli mode that you see over here, the gray values, I can actually reduce significantly the amount of material. I make a zoom here. So in this zoom, okay, let me go back. In this graph, I have the frequency of events. In this graph, I have pH values for each of the hundreds of endosomes that you have over here. What you see over here is that most of them are primarily in the acidic region, you could say below 7. And there's a very, very small fraction in the order of 2%, and I will come back in a few seconds why this is important, in the order of 2 to 5% that they are neutralized, hence pH above 7. And when we start zooming into there, we can see that the number of neutralized are dependent on the presence of PFFs. And then you can see here that when I add also papilli mode, which is a molecule that drops a fifth kinase in the cells, I can eliminate or reduce drastically the fraction of molecules that are actually being neutralizing. In a way that shows that if I am to summarize all this in a single slide and not overkill you with all the biology of the system, is we can observe in real time preformed fibrils and enter in the cell. We have identified that they enter through endocytosis. We have identified that primarily at the late endosome, okay, I'm losing the wrong finger, that at the late endosome, you see that you have a neutralization. And furthermore so, we can revert this neutralization with small molecules over there, as you can see over here. So that was a readout that integrates quantitative for the imaging, cell biology, and a lot of the machine learning analysis that you can see on the left that is coming out from my group, that for each of these data points, we can actually in real time extract all the information that you need to have. And now that we have extracted this information, I was like, but can we actually observe the kinetics of the process of protein aggregation in real time? Can we extract this layer of information? So then I had an extremely talented master student, Steen, who published this. That's his master thesis. It came out in Nature Communications in 24. And Steen, what he developed is what we call CMOR, is a tool that allows us to see this process as they happen in real time, and B, analyze this. So what you see here, and this is collaboration with Novo Nordisk that paid a few of the students in my group to develop the super resolution method that you see here. So what you see here is a microscopy setup that each time a chromophore comes in, as the protein aggregates, you can see the seeding and the protein starts forming a fibril. And either the fibril can continue as a straight line or a fibril, or it can start branching as a tree. You can have the first branch that you see over here, and this branch is branching more, and this branch, as you can see here, is continuous branching. It's pretty much the fractal, as we call it in physics world, that it shows how one branch leads to, for example, to 2 branches, and each branch leads to 2 branches, and so on. And in certain cases, you can see that it doesn't really grow like a tree, but as you can see here, it grows like a sphere. We call this a spherulite. Like a bush, if you may call, it never becomes a real tree. And in certain cases, it primarily stays like as a fibril. And as I'm going to show you, the conditions by which and the concentration and mutations, etc., will have a profound effect on the actual shape. So we firstly developed a super resolution method that we can observe this in situ and quantify the growth rates, etc. And this was extremely interesting because that's when Steen came to the group. And Steen was like, but we do this with quantitative imaging, and it takes us a few days to analyze all this data because we have to find the particles, click on them, I'll make something better. So what he made is what I'm going to try to explain in a very simple words. We call it CMOR, which is in essence is a fingerprinting of the protein clustering and aggregation. And I need to stress the word clustering because we initially applied it for aggregation data, but we actually have shown in this paper that it can be applied for clustering or for extracting dimensions of nuclear pores or whatsoever. So the whole thing is a multi-modular machine learning framework. And I'm going to explain it in terms, and then I'm going to explain it in English. So, the first module here is a clustering module, which is a density-based clustering. It starts identifying all the elements of that are forming, if they belong to the same cluster or multiple clusters, and if these are meshed. Then the second, we have a temporal refinement module that you see over here. This temporal refinement module, what it does is it outputs the identity of the subclusters. It does a time-dependent clustering, and then it outputs if the cluster branches, etc., and when this happens. Then this becomes, and that's what you see over here. And then what happens is this model outputs all aggregate structures like that you see here. And this becomes the input to the fingerprinting module. And the fingerprinting module, it takes 4 generic features that describe the aggregation process. It's the circularity. Is it circular? Is it long? Is the geometry? Is the network, how dense it is, and the symmetry of the molecules. And then based on this, it outputs all the characteristics, and it predicts very early, somewhere here, what will be the final structure. If I come now and say this in English, you're sitting in your garden and you're seeing in a time lapse some of your, tomatoes. I don't know. Most of you live in a civilized country that tomatoes can grow. They don't grow in Denmark because it's too cold and there's no sun. So you can see your tomatoes growing, and you can very clearly see that in some cases they start and then they branch, and then each of them branches very clearly. And then you have another type of plant, I don't know, courgettes, that they grow in a different pattern. You very clearly, somebody that has done this work looks at them, is like, okay, these are tomatoes, these are cucumbers, these are courgettes. Why? Because you can recognize the leaves, you can recognize the shape. Yes, very early in the process. And what we made here is a machine learning framework that takes us into all these early steps and then radically outputs the final structure. And when I say radically, I want you to mean rapidly, because I'm going to show you the aggregation process and how fast this happens. The aggregation process starts here. In the beginning, everything is black, because the model doesn't know what they are. I'm going to replay it because then you can see when they start changing colors. When they change colors, the model is able to predict the final structure. So what you see here rotating, X, Y is the coordinates of the particles as they grow, and Z as they go on top is time. So you can see how they're spreading. I'm going to replay the movie. Oops, sorry about that. I'm going to replay the movie. Right there, you have 80% of your elements. In the next time frame, you have 90% of your elements that are already identified. Pretty much as you did, okay, this is a tomato, this is a cucumber from the first leaf. That's what the model does. And in doing so, it accelerated our analysis 3 orders of magnitude. And it allows us to predict, and I think that's the most important. One thing is it accelerated by 3 orders of magnitude. The second is we can now go into the aggregate, even if they're not fully formed, and say, oh, that's going to form that. That's going to form that. Yeah, and that's a profound implication, as I'm going to show you in the next slides. So now we made the tool to a super resolution method to observe in real time as the aggregates growth in these in vitro systems. We have a machine learning framework to analyze this data. And then somebody might say, but can you actually see when your cells are dying? Can you actually identify with a click of a button when this is happening? That's another process that we did. And because a big part of my group is machine learning guys, they don't like easy tasks. They're not inspired by easy tasks. So they asked, what's the most difficult type of cells that I can segment and do this behavior? And the cell biologist of the group said, oh, endothelial or epithelial cells. And they were, all right. So what you see here is we have made a monolayer of epithelial or endothelial cells. These guys are endothelial. We have done exactly the same for the endothelial cells. We put them on the microscope, we label their nucleus, we label their membrane, and then we start administrating different types of ligands or different types of fibers or different types of final structures, because now we know how to make the final structures. And when you talk to the biologist and you say, oh, my cells are stressed, oh, my cells are dying, the question that we were always asking is like, how many of them? What's your metric that your cells are dying? How do you know that your cells are stressed by a certain element or a certain molecule? And the answer is, but you can see them. And then, of course, as I said earlier, you can really separate tomatoes from potatoes, but how fast? And can your son does that? So then we were like, all right, let's make another fingerprinting module, which I'm not going to go through all the details. This is still in review. We call it now cell morphology fingerprinting. And etc. It takes all the feature organizations of the cells, their shape, their size, the compartment, the intensity of this, and it makes a mask of 41 features. We do a dimensionality reduction out of this, and we extract what are the features that are important. And based on this, and now this is the interesting part, because now you can go out and identify different ligands that have a profoundly different effect on the morphology of the cells. So the cells are dying, but not all with the same way. And these experiments are done for different ligands that we can bind on cells, but they can also be done for different types of protein aggregates. And now you can see here for the non-machine learning lines, this is a confusion matrix that says how accurately you identify this condition from another condition. Yes, and you can see that you start reaching 90%, 85, 86, 90, and 96% accuracies on all cell types that you can recognize the effect based on the ligand that you just fed them. Now you might say, okay, that's cool, Nikos, but can you do this on cells? Are the different types of aggregates that we discussed earlier of the insulin or the synuclein aggregates are having a profound effect, differential effect on mice? So then we took some of the aggregates that we made with insulin this time. We're going to Novo Nordisk. Novo Nordisk is paying a few of the PhDs and postdocs in my group. We did mouse studies, and we took the types of spherulites, the dimensions, the long ones, the branched ones, etc. And we started evaluating on mice and human-derived immune cells what is the effect. And you can actually start, because, and I think this is important, because we had tools, and I realized that there's so much information that I didn't give you, and I apologize for that. Because we had tools to extract the different types of structures, we have now also tools to direct the formation of a certain type of structure versus another structure, because we now have the mechanism. So when we start feeding this in mice and cells, we can see the immune response is dependent on the dimension, the geometric morphology, and the content of the spherulite, and also see which of them are more immunogenic. And why is this important? And why did Novo Nordisk engage on this? Because it turns out that if you take insulin, hopefully none of you needs to take insulin formulations, if you mistreat it, either because you have it out of the fridge for a lot of time or you shake it vigorously, it starts forming a cloudy solution. This cloudy solution are particles that are above the diffraction limit of light, a few hundred nanometers. And it turns out that when you are in this regime, that's what you see here. This method allows us to evaluate the immunogenicity of particles that are in certain diameter ranges in cells. All right, so now you got all the concept. I hope I have convinced you that we can observe the aggregation process. We can control, to a certain extent, the aggregation process. We have developed the analytical pipelines to control it, and now we have a very good understanding of how different types of aggregates. This is published for insulin, and we're doing this for synuclein, has a different immune response in mouse and in humans. And now this is interesting, but now you might say, can we utilize this information to actually go out and evaluate the delivery of pharmaceutics to target this? So this is a part of another project that I'm involved. It's called the Nano-Science. Is €6 million program that is led by Martin Larsen. And the whole idea is to deliver therapeutic antibodies in the brains of mice and across the CNS, as we call it. This is with Professor Larsen, and he is 50% of his time seeing patients and 50% of his time doing experiments on mice. You realize that when we say he's doing experiments on mice, it's not him actually, but his students, but you understand the concept. So the whole idea is that we take a mouse, we cut a little piece of the head, we put a glass slide, we stick them on a 2D microscope, on 2-photon microscope, and before you say we are cruel, these are a stress test on the mice. They're perfectly happy, the mice, and we can actually see their responses, how fast they walk, how happy they are. So we have a very good way to evaluate all this data. And then we deliver lipid nanoparticles, as I will come later on, loaded with material, either genetic material, as I will tell you later on, I will touch upon, or with therapeutic antibodies. And now we can see actually inside the brain, we can start seeing which of them are inside the arteries, which of them have managed to release inside the brain. And as the movie plays here, you can see blood, you can see a lipid nanoparticle that now penetrates and penetrates through the brain barrier and goes to the brain parenchyma, and you can follow this for some time. This is fantastic because that's the first time that somebody can do this on a mice. But then the question that you might be interested in is like, which cells does it get stuck? How many of these nanoparticles enter the cell? How much of the material do I manage to deliver inside the cell? So now we use another type of microscope to actually start accessing this layer of information. So this is a lattice light sheet microscope that I have in my group. And these microscopes allows us, these are neuron cells differentiated, and we can actually go out, not just at the mouse brain, but now we can zoom in individual neurons, as you can see here. And if I zoom into this region, you can see the soma, you can see the action of the neurons, you can see when they're forming synapses with neighboring neurons. You have a quantitative imaging of the whole process. Here I have labeled the neurons with the GFP and the membrane, and the red is actually interesting because these are lipid nanoparticles that I have done two works. I'm going to focus on one of them today. One is that we deliver therapeutic antibodies, and we also have tested on the outside with targeting motifs. And most importantly, I believe we're on one of the first tools that we deliver efficiently siRNA or mRNA, like a vaccine in neuronal cells. I'm not going to talk about this, but this is part of the big center that I direct. Here today, I focus primarily on antibodies. And now you can see, excuse me, you can see here, I really zoom in and I'm looking at the soma of one neuronal cell. You can see the axons over there. And now I can see that now I have labeled the transferrin receptor. I have specifically targeted the transferrin receptor of this by my antibodies because I only see on the soma the signal. And we can go in and see this process in real. I can zoom in, I can do a fly in, and you can see that if I change the contrast, I now see that I have nothing on the axons and I only have something on the soma, which is very characteristic if you're targeting the transferrin receptor. So you can see this in real time. And now you might say, okay, this is cool. Oh, this goes fast. You can see this is cool, but can I actually extract what's happening in there? Can I actually find out how many lipid nanoparticles went inside the cell? How many of them released cargo, either the therapeutic antibody or the genetic material? So then we go back to the live cell imaging, and these are, now I'm going to focus on two of the neurons here that are connected next to each other, and now each of the spots is a lipid nanoparticle loaded with antibodies. And when the movie plays, we're coming back to what I earlier told as a 4D. Now you can see the particles. This is lipid nanoparticle. This is brighter than this, simply because it is larger in its size, and this is brighter than this, also because it's larger in size. So the color, the brightness signifies dimensions. The lines that you see, probably they are more clear here in this color. The lines that you see over here correspond to the pathway that these nanoparticles follow as they enter the cell. And you can very clearly see this guy is completely stuck on the membrane. It never entered the cell within the 10 minutes that we are looking. It could be that it enters later on, and we have tools to analyze this. And you can see other guys that they're moving very fast. Now the movie plays backwards, so it appears that it becomes shorter. But if I play it the other way, if I started from there, you will see that the particles that are inside the cell, they're moving extremely fast. So out of this, you can actually, your eyes can really tell, oh, but this is stuck. This is inside the cell. This is inside the cell. But you don't want your eyes to do that. You would like what we call one-click biology. You would like very much, with a single click of a button, to have all this information. So you would very much like to look at the data and extract this. So that's how we do it. We use lattice light sheet microscopy, and this is something invented by Eric Benzik. And it's a few copies in the Janelia Institute in Harvard. We have one in Denmark, and there are a few copies in other labs as well. And now this allows you to take volumetric imaging of the entire cell, and you can find your particles, you can localize your particles, find the center, extract the X, Y, Z coordinates, which is what GPS does for you, and get a trajectory. And this trajectory is really what Google Maps does for you. Every morning when you say, can you, or your app that counts your steps in your phone, that you say, count my steps, it very clearly knows if you're walking, if you're running, if you are in a bus or something, because of the directionality of the motion, etc. And that's what you see over here. What would be really, really, really cool is that when we look at this motion, yes, if we could actually say, oh, I see this type being a little bit different than this. And that's probably because my guy is stuck on the membrane and this guy is inside the cell. It would be absolutely cool, which is, by the way, what your phone does and gives to Apple and Google. And my phone does and gives to Apple. All your spatial coordinates and your motion types are utilized to extract this information. So it would be really cool if from this you can go all the way to that. But this is not straightforward because you need to be an expert. And ideally, you would be like to see a type of motion, which I will explain in a few seconds, and utilize this to predict identity, position, function, etc. And if I want to say the types of motion that you can have in the cell, I can very broadly and generically characterize them in four. One of them is this directed motion. Directed motion is you're driven by a molecular motor and you're going almost on a straight line. That's when you are in your car and drive on the highway, clear. Then you have a confined motion. I am in a confined motion right now. I have half a square meter that I'm moving in and out because I want to see what's happening in the screen. Then you can have the subdiffusive motion. And this is the motion that you have when you are in a party. When you are in a crowded environment, you want to go straight, but you're bouncing on somebody. And then you do a small detours and you're moving, but you don't move the speed that you would like to have. And last but not least is the completely random motion that you can go in everywhere in all types of directions at any given time. It's a stochastic variation. So I know this sounds very techy, but this is the types of motions you could have in the cell. Because, for example, confined, you are inside the endosome. Subdiffusive, you are in the cytosol because you have, it's a dense environment. You're bouncing in other types of molecules. You don't move straight. This, you're driven by a molecular motor on actin filaments, on endosome. And this, you're in solution. You're a Brownian motion, as we call it. So it's completely random. So it would be awesome if we could extract this information just by looking at the data. This is this is the work of another extremely talented student of mine. And Henrik in his, actually that was, the work was done in his bachelor, if I say it in Greek, in Diplomatikito. And then halfway into his master's, he got his PNAS paper. And he made what we call a diffusional fingerprinting. And what is a diffusional fingerprinting? Is a unifying machine learning framework for analysis of anomalous diffusion. And I will explain in a few seconds what it does, complicated and then in a simple language. So as an input, we take the output of a tracker is the set of X, Y, Z coordinates that you see of all here for each particle. Then out of this, you extract a few features. We call it, here it's 17 in the beginning, diffusional features that describe the motion. How fast does it go? Does it go in a straight line? Does it change directions all the time? For how long was going fast? How many times did it go fast and slow? How many times did it stop? All these type of features that describe the motion. These 17 features were projected in a 17th dimensional latent space, out of which we using a logistic regression, we collected the first 3 features with a mechanistic significance, and we collapsed this here in a 3-dimensional map. And now on this graph, you can see here, out of the 3-dimensional map, you can actually see the significance of all these figures, the sharp values. Okay, I'll say this in English now. So what we did is, imagine you're looking, as I explained earlier for the garden, you're looking at a person walking down the street, and you're like, this person is wealthy. And it probably is because he's driving a big Audi Q3 or something like that some people drive, or he's driving, or he's having a nice suit, or he's having an expensive watch. Yeah, and then you see another person, and now it's her bag, it's her hairstyle, it's the way she moves. What your brain did is it took all these features and it valued them, and then it said for A, B, and C reason, and this can vary from situation to situation, but for these 3 reasons, I made this conclusion. That's exactly what this machine learning framework does. It takes all these features and it values their significance. And because it's not a neural network, but is actually it's based on the diffusional figures, it actually outputs also information, because we know, we have information of mechanistic significance out of this. So that came out in 2021, and that was fantastic. But then we were like, okay, that allows me to identify that rich people, wealthy people move differently, but can identify when things change. And this is when another guy of my lab came in, and Jakob is now a postdoc in the Boston area as well. And Jakob got developed, and this paper came out in 4 months ago, 5 months ago, something. And Jakob, what he did is we call DeepSPT. And what is the DeepSPT? Is an end-to-end machine learning framework that takes motion and doesn't just output behavior, but outputs identity. It says, oh, Yannis Sidiropoulos is the guy that I saw with his car. Just by looking at his car and by he moves. Oh, Maria is this person. Yeah. Why? Because it takes the data, and I'm going to show you how it works. Very simple. I'm not going to go through the details because I'm going to explode you with all the analysis. But it takes, the first thing it does, it takes this heterogeneous motion, it segments it, then it gives the data to the diffusional fingerprinting module that I just explained a few seconds earlier, and this outputs all the characteristics, and then you have a classifier that says, oh, this motion means that. So in essence, if any of you has a smart watch, that's exactly what your smart watch does. Your smart watch knows if your hand is moving like this because you're walking, or because you're dancing, or because you're sitting. And that's the first part. The second part, if you're walking or running, the acceleration will be different. And that's what the second thing does. And the third thing does is somebody needs to train the model. Yes? So somebody took 100,000 people that were running, 100,000 people that were walking, and saw all these patterns of motion, and it trained the classifier. Okay, take this, not for 1 meter person moving at the mini time scale, but for a 50 nanometer protein or a 5 nanometer protein that moves at the millisecond time scale. That's exactly what this method is. So that's what Jakob did. And how good does it work? I think I'm running almost out of time, but we also start 10 past, yes? So I'm at 40 minutes now. All right, so then I'm not going to kill you with all the details. We use this method. Here is individual cells. This is a rotavirus, which is a virus that causes diarrhea. And these black lines that you see here is the trajectories of the rotavirus as it enters the cell. And I took this trajectory over here, I expanded it here, and the blue line is the actual trajectory of the virus. The red magenta is the actual trajectory, the motion of the genetic material of the virus. So what you see over here at this time point, over there, the genetic material is released from the virus, exactly. And by the time it's released, you lose it because it moves very fast inside the cell. And below is when DeepSPT predicts when it was released. But DeepSPT doesn't look at the red, cannot access the red. The model is only fed with this data. So it picks up only the blue, and it picks up ever slightly variation on the motion. It says, aha, right there, you have a release event. How accurately does it do it? It reaches up to 85% accuracy without the need of labels, guys. And that's the important thing. It rapidly outputs the release event without having to label the genetic material because it can recognize the motion. And I think it's really important, the next statement I'm going to make. These data were acquired in Tom's lab while I was doing my sabbatical there. And the reason we did all this was because while I was there, the guys would need 2 to 3 weeks to analyze the data. So you go into the microscope, you acquire some data, and 3 weeks later, you're like, I should have double the concentration. Yeah. When we analyzed this framework, we, from 3 weeks, made it 500 milliseconds per trajectory, 6, 7, 10 minutes, offering practically live analysis, 6 orders of magnitude acceleration. Yeah. This is massive because it's a mind-changing game when you design experiments or when you do screening events of mutations, etc., that you can analyze them so fast. And then I'm going to show you that we can actually directly observe this release event. So this is the virus. In here, it plays the genetic material. Here, I have overlaid them for clarity. I displaced them by a few pixels so you can see them. And you can see I'm looking at the virus. I'm looking at this cargo right there. I'm going to lose my cargo, but I still look at the virus. What does that mean? The virus opened up, released the genetic material. Okay, so we captured this on the microscope. But the most important element here is that the color code here, from magenta, it turns blue. And that's the machine learning that just looks at the ever slight variation in the motion and says, oh, right there, you have a release without looking at the cargo, just looking at it. All right. I think I'm almost done there. And you realize that I don't do it in a single particle. We do it at 1,000 or 100,000 particles in a day. And then you might want to say now, okay, that's cool, but you showed us that virus. Can we actually do exactly the same for antibodies that are inside the cell? This is what we do. So we track the antibodies, we track the endosome, and we also see when I start losing some antibodies inside the cell. And not only that, but then I can start comparing different antibodies, which I will not disclose their names. I will explain in a second why. And now you can go out and say, antibody A versus antibody B has more internalization events, more coming inside the endosome. But now, B has actually, A has, even though it has less internalization events, it has more antibodies in each internalization event. And then we can see how many of them are losing their cargo in the cytosol, releasing the cargo in the cytosol. And this is extreme, and you can see some of the publications of the group on similar studies for the quantification. And what is extremely interesting here is that these two ladies work on different antibodies, and they're frenemies within my group because they are paid by 2 different companies. So they call their antibodies Agent Smith and Neo and something like that because they cannot disclose the original name. So they share methods, they share results, but they don't disclose what antibodies they work on. This is extremely interesting. I think in the interest of time, I will just say one thing here. It's a very complicated slide, but for anybody of you that does cell biology, it would be cool if you could actually inform, or if you understand what type of compartments you are looking inside the cell without having to label the compartment. Yeah, you would like to know if something is on the membrane, if something is on the early endosome, if something is on the late endosome, lysosome, in the Golgi, in the ER, etc., etc. And that's what I'm displaying is that I can really tell early endosome from late endosome just by the machine learning framework that I just described you. And if I try to use any conventional methods, they're completely indiscernible, but I can reach an accuracy of up to 80 or something % just by looking at this data. And I'm going to finish with the last story, because I mentioned the word one-click biology. And I'm going to show you the next paper we're really about to submit. And this is really the concept of one-click biology. It's a large language model that you talk to it, and it does it. So imagine if you got the cells and you say, find my cells, and find the particle that they are inside. So it goes out, it localizes the cell, it finds the more spherical cells over here. It finds the cell in one plane of the microscope, and we do multi-plane, obviously, because we do 3D microscopy. Then even though it found it in one plane, it can still recognize it at different planes. So it's like looking at the area outside of your house. You can find your house, you can find the garden, even though this is on a hill, etc. And not only this, but as the cells moving around, it can still track the cells, the shape as they change or they move in the XY. And most importantly, it can go inside them and say, okay, let me now find all the particles that are moving inside the cell. And based on the tools that I show you, I will extract their identity. I will extract how many proteins are there, how many of them are released, or how my aggregation grew, etc. All right, this is similar to what we say in my group. Is like, you see, this is Mission Impossible. You see the airport, they say, go and find this guy. It starts tracking all the people in the airport, and it finds a certain person. That's exactly the method you see over here, but not at a meter, but not in nanometer time scale. All right, I will stop here. I think I'm running out of time. And this is a fantastic team of researchers. It looks like a big group, but 1, 2, 3, 4 persons have graduated, the rest are still here, and then we hired some more. And then it's a group that is really an incubation hub of ideas, because you have people that are computer scientists with machine learning, they're cell biologists, microscopists, chemists working together. And then we have software engineers that take the code that the machine learning writes, make a software for the rest to use. So it really is a conveyor belt of ideas and how we move and really bridging new barriers on a daily basis. I will stop here, and I'm super happy to take questions, guys.
Thank you, Nikos. Really impressive work, and I think it's mind-changing for many of the cellular work we're doing, either with aggregates or antibodies or even exosomes on or delivery of drugs, and that's something important, because we can go back and really understand what method or what particles to use to deliver stuff. Or, I mean, you didn't say too much, but I know you mentioned it once. You also can do it with activity of enzymes or that not only follow the release, which is even really huge breakthrough in the field, but you can also see enzymic activity, correct, and how it changes inside the cell.
Yes, one catalytic cycle at a time. So we can resolve that.
Yes, for sure.
On this audience, but yes, we have many publications on a lot of work on that. Yes.
I receive messages from people that how enthusiastic they are. I don't know if they're afraid to ask questions, but guys, do we have questions?
May I ask something, Yanni?
Go on.
Nico, thank you. Are you sure a chemist? I'm not sure that you are a chemist. Okay.
So, I am by training.
Okay.
I was a chemist, if I want to be more correct there.
Right. So perhaps you said that already, but it was very interesting. When you detect all these details in movement, in space, and in type, can you do it simultaneously for 2 or more molecules?
For a million.
For a million. Then you can track also all the interactions and what is changing upon the interaction of 2 or 3 molecules.
So you're asking if 2 elements are coming together and
they have an interaction, and then the structure of the function is changing. Can you detect that? Can you predict that?
The short answer is yes. So we had a paper, I didn't talk about this, for CRISPR-Cas12. And there we were using single molecule FRET. So we had the protein, and we could, and FRET is the spectrometric ruler that gives distance information. So you actually can observe the opening and the closing. So we had done this, and then we had labeled guide RNA or mRNA, all the molecules that are relevant. So we could see how the closing and the opening, we could see in real time the opening and the closing, and how this is affected by the time of molecule you have there. So we combined with cryo-EM, we actually not only solved the structure, we found 4 different structures, and with a single molecule thread, we said, oh, but this structure goes directly to there, and this structure talks to there, and not only, so we found the sequence of conformational transitions, and we actually output even the energy barriers. So that allows us to combination of EM and single molecule thread to plot for the first time the complete multidimensional landscape of this protein and do that. So that was in vitro. Now, and that is with Guillermo Montoya. Guillermo is one of the leading authorities on the CRISPR and cryo-EM. And now we are doing exactly that, but now inside the cells, inside bacteria. Which you have motion as well, not just in a mobile particle that opens and closes. So you're opening and closing, you're moving around, and all this is dependent on the interaction with other molecules. So we're doing this as we speak, if that's what you asked, Yanni.
Thanks.
Nico, can you stop sharing this so you can see all the panel? That's very good.
The real impressive in this, Nico, is that it has no limits. I mean, you can find so many things.
Exactly.
There's a limit on the data.
The only limitation, Nico, is to find molecules to label different parts that you need to find. That's the only limitation, which is indeed, it's not a real limitation. You can find it.
And the analysis.
The big limitation is the analysis, because you need a lot of data and a lot of AI support. But since Nikos has all the pipeline, I think that's not... Savas is also waiting.
Yes.
Thank you for giving me the opportunity to ask a question on this excellent work and very impressive and inspiring. Thank you, Nikos. Fantastic work. I wanted to ask something about the sequence of events of the synuclein experiments that you showed to us. So synuclein enters, then it oligomerizes and ruptures the membrane due to oligomerization, somehow damages the membrane due to the increase of the aggregate size, which then neutralizes the pH. Is this the sequence of events? And if this is then... Okay, and then why is the percentage of the vesicles that get damaged not higher than what you showed? It's a small percentage of vesicles, so why not all of them, and why a few
You're
of them?
touching some very key points, Salvan. There are multiple questions there. Let's start with the last one, because then, you two, I think spending some time with you, honestly, I got some Alzheimer from him, so we share the same problems. So the first question you asked, because I will forget the first question. The second question you asked is about the low percentage. So the field of delivery of oligonucleotides, for example, is actually hampered by the limited escape of the endosome. So we are now, I didn't talk about this at all. We will release in bioRxiv soon a massive paper and then send it to an interdisciplinary journal that we actually quantify the fraction of endosomes that rupture and how this results to the release of genetic, viral, whatsoever material out of it. This, I'm telling you, is in the order of 5% or 1%, depending on the composition, So it's a very small number. And it turns out, and that's what came out, and I apologize if I didn't make it clear in the first paper that we had with that I started. What we see is that endosomes transiently, stochastically, if I use biophysics word, sample a ruptured state. And when they do so, it appears, that's what we say in this paper, when they do so, synuclein from solution comes in to the preformed fibrils, and then it starts growing. And eventually, it destroys the whole thing. Yes? Okay. But it's this stochastic, random, and infrequent opening that drives this whole process. And by the way, I was talking with Tom today on our weekly meeting, because I'm still part-time there, Tom Kirchhausen from Harvard, and they just sent yesterday exactly the same for tau on bioRxiv. So it's online now. So it seems that it's a unifying concept that there is a transient rupture of all of this, which occurs in the order of 1 to 5%. And this is work in progress, but and that drives the formation of higher order aggregates in there. That's at least the data that we have up to now.
But you had the synuclein from outside, is that correct?
Yes.
So can you exclude the possibility that internalized synuclein oligomerizes in large oligomers And in certain vesicles, these are enormously many components that are oligomerizing that rupture the membrane due to the fact that the protein polymer disrupts the membrane as it increases its size in length.
For that to happen the opposite way, you would be able to observe synuclein aggregates away from the earlier late endosomes. And this quantitative image analysis did not show that.
Okay.
Or, just to put it Sava, because it could be like we missed something, yeah? I mean, when you look at the 10,000 particles that what are there, we couldn't detect to any meaningful significance, at least that is statistically significant events that are outside. It could be that some of them do escape, but the vast majority of the elements, we found them colocalizing with early and late endosome.
Okay, thank you. Thank you so much.
Yeah.
Can I ask something?
Go, Vaso, go.
Okay, hi. I mean, amazing work, of course. Following up on Sava's and what we about the alpha-synuclein, have you done same or similar studies for A-beta aggregates? We are working on Alzheimer's disease, so it's something that would be interesting.
No, actually, no, but we are, as I told you, I'm a part of a smaller consortium. So it's only 3 PhDs of the group that actually work with delivery. It's what I talked primarily today. And there the concept is that we will have to do A-beta too. However, I need to be very honest, we haven't done the aggregation process of this. It's like we have the tools, but we haven't studied that specific protein. So we would have to do the aggregation process in real time, as I showed you. The machine learning work should work on this and then add them on cells and see the whole process. It will take us some time, but that's the scope of that project, to observe this in real time in cells, in neurons,
yes,
and ultimately on brain, and see if we can reverse it or inhibit it, either by siRNA, that you downregulate the production of proteins, or by therapeutic antibodies. That's the consortium that I am.
Okay, thanks.
Any more questions? Nikos, do you have any problems of clocking or being together like 2 or 3 molecules for different reasons? That if you have the aggregates, how easy is to identify the single molecules? I know your microscopes has very good resolution. However, when they're together, it's how easy is to identify single molecules and separate it from oligomers. And the first question, if the answer is easy, or let's say relatively easy, then did you see patterns? Why I'm saying in all proteinopathies and all these aggregates, so if it's tau, amyloid beta, or tau, there are shifts on different stages of neurodegeneration, let's say in general. So there are mechanisms, cellular mechanisms that they push from the single molecules to small oligomers, that they're bigger oligomers, and then there are swifts to even bigger aggregates. And depending on where it moves, on different biological actions, for example, if there is an autophagy dysregulation on the endolysosomal pathway, cannot throw them out through exosomes or whatever. Did you see any of these subcategories, or if you are able to, let's say, categorize in subcategories of smaller, bigger, or next to the nucleus, far to the nucleus, late endosome more?
That's a good question.
Is it easy? Yeah, because even for the project we discussed with CMOS, that with different stimuli, but that is one of the ways to show in the next step, okay? But that's why I'm asking you whether it drives to one or to the other direction inside the neuronal malfunction or using neurodegeneration. And what is describing to us in the time frame you're describing, it makes a lot of sense that neurodegeneration is a long process that takes really years to happen in human brain, okay? And in mice, in a different, age scale, but it makes very much sense. So my question is, if you can see this different localization, different type of aggregates, and by time, what is the time you can monitor or follow these cells? Induced IC, iPSCs, induced neurons, or whatever. How much you can follow them in time? I know the data you're creating is go to Labrio.
No, but the first question is if I can separate the single from multiple. I mean, spatially, you cannot, because they are below the diffraction limit of the microscope. So you cannot tell them apart if it's 1 or 2 or 3. However, the intensity of these guys will be completely different, because one of them emits X photons to 2X, 3X, 10X. So you can extract this layer of information, albeit this information doesn't yet give you, if this is a linear or a sphere, it doesn't give you information about the geometry. That's why we developed the super resolution method to extract that information as well.
Okay.
The second question that you asked is if I consider spatial localization and correlates, if I understood correctly, spatial localization with copy numbers of proteins or aggregate morphology and size. I didn't even think of that.
Really. We can discuss.
But the information is there. For us, the information is there.
Okay.
I mean, the data is there. So
that's what we'll start discussing between you and Simos. That was the Simos stuff we discussed.
Yeah.
Okay.
So
good.
It's going to have to be something downstream, but it can be done. And we actually do, for example, see for lipid nanoparticles when we deliver in cells either siRNA to knock down or certain proteins or mRNA to express certain proteins. We see very distinct localization patterns with escape. So this is something. This is work in progress, but we see these things. So we can quantify these things, albeit we haven't done it for the system you asked.
Okay. Thanks a lot. Do we have any more questions? Then if not, once more, we would like to thank Nikos for this great and very inspiring work, as well as the presentation. And the fact that you make it easy to follow, it's also something important for us, because in the audience there are different people from different backgrounds. Thanks again. I hope this Zoom will start to think as a lot of questions and synergies between different people from here and both parts of the Atlantic, Europe and the States. Thanks a lot. And for the rest, we will see you in the next month with the next HIAAD seminar. Thanks, Nikos.
Thanks, everybody. Have a nice day.
Ciao, man.
Γεια σας.
Thank
Γεια
you.
χαρά.
Γεια σας.